Well, hi everyone. I talked about the microscopes, fluorescence, confocal microscopes, and what have you in the first part of this series. Today I'm going to tell you all about the experimental techniques. I'm going to concentrate on experimental techniques for confocal microscopy, but at the end of the lecture, I'm going to talk about electron microscopy as well, just for completeness sake. So experimental techniques in advanced bioimaging mean you're at the microscope. What do you do with it? How do you make scientific discoveries, basically? So we'll call these quantitative imaging techniques. In other words, techniques that let you generate data that you can measure so that you can determine, oh, the rates of protein movement within cells, the rates of organelle movement within cells, how things inside of cells interact. And I'm referring you on this slide to a paper that we wrote many years ago now called Quantitative Fluorescence Microscopy, From Art to Science. In other words, after watching the first part and getting your hands on a microscope like this, you should be able to make beautiful images. How do you go from making beautiful images to making images that give you useful measurements about biological phenomena? This is a big paper, and there are lots of pictures like this in the paper illustrating different techniques. I've asked you as part of the Bio 6001 cell biology course to do an essay for me on experimental cell biology, and I've assigned a list of topics that include things like FRAP, which is indicated here, FRET, which is indicated here, and bimolecular fluorescence complementation, which is indicated here, and I'm going to go through each one of those techniques in this presentation. So using light microscopy to experimentally gather data on cellular function. And people started coming up with techniques years ago, and we jokingly called them the F words at first because they made up acronyms that indicated what the technique was. FRAP, fluorescence recovery after photobleaching. FRET, fluorescence resonance energy transfer. FLIM, fluorescence lifetime imaging. And then we departed from the F word tradition. TIRF, total internal reflection fluorescence. Photoactivation of some fluorescent proteins. Multiphoton excitation. Deconvolution. And then there are super resolution techniques like ARRI scanning. We'll call that a near super resolution technique. Stimulated emission depletion and structured illumination microscopy. So I will talk about ARRI scanning instead later on in the lecture. And I'll mention most of these things here anyway. Fluorescence recovery after photobleaching is something that's been my research go-to tool for the last 15 years. An absolutely amazing technique designed to measure the rate at which things move around inside of cells. And I don't just mean the organelles, I mean proteins. If you look at this picture here, this is a sheet of plasma membrane looked at from above in a cell. And the plasma membrane is impregnated with a protein that's anchored. It has a transmembrane domain in the plasma membrane, and that protein itself is fused to GFP. If you go back and look at the first part from last week, you'll see me talking about how you fuse GFP to a known protein to mark a subcellular organelle. So in this case, we've marked the plasma membrane with green. Now, if we target a laser right down on the center of this cell here, and we turn the laser up high, just for a quick pulse, we can bleach a region out. Watch what happens when I play the movie. To see the little square appear. I bleached a square region of interest. And over the course of the next minute or so, in real time, the video is speeded up, you'll see that the area that was bleached fills back in with fluorescence. Well, the GFP that you bleach doesn't recover. It's dead in the water. It's been destroyed. What happens is the proteins that GFP is fused to diffuse away from the bleached area, and new proteins that were outside of the bleached area diffuse in, and they fill it back up with green. So this diffusion is a characteristic of membranes, proteins in membrane, lipids and proteins in membranes. In this case, what I want to do is measure the rate at which that protein, the plasma membrane protein, moves. So we do the FRAP experiment, and at the end of the experiment, we measure how long it takes the fluorescence to recover in the bleached region, and to what level it recovers within the region. And as well as measuring that region there, you'll see that there are 3 other regions that are indicated here. I also measure the fluorescence within those regions as a control in this experiment. So if we look at a graph like this one here, this is fluorescence intensity on the y-axis here. And this is time on the x-axis down here in seconds. So the fluorescence intensity, the mean intensity of the green fluorescence in regions 2, 3, and 4 stays level all the way across the experiment. It doesn't change, and you can see that in the movie. If you watch this region here, it doesn't bleach, it doesn't get dimmer, it doesn't get brighter. But in the region that's indicated as number 1 that's bleached, it starts out bright. Here's the laser pulse here, the fluorescence drops right away, and over the course of the next 100 seconds or so, you see that the fluorescence increases again. It comes up. It doesn't increase to the 100% level that it started at, but it increases to some level, and we measure that level there, that change, and we call it the mobile fraction in a FRAP experiment. We can also derive things like the half-time of recovery, which tells us how fast a protein is diffusing. So a very, very useful tool for measuring protein kinetics within membranes. Talking about the study of membrane proteins, just as a little background, here's a protein that's equivalent to that one that I was just showing you. It's embedded within the lipid bilayer of the membrane, and it's got GFP attached to it. And the recovery into the bleached region in the FRAP experiment is because these proteins diffuse around within the plane of the membrane, within the bilayer here. There are all sorts of ways that proteins can be attached to membranes. This one is a single, or in fact, if I remember correctly, it has 2 transmembrane domains, and it's got GFP attached to it in here. There are lots of other ways. I'm not going to talk about those at the moment because it goes a little bit off topic. We did an experiment, though, looking at a protein that was roughly equivalent to the one I showed you in the first place. We bleached this circular region in the membrane here, and over the course of 60 seconds, this protein diffused back in and pretty much filled the bleached region. This protein was called GPA1, fused to GFP. So if we go look at one of those FRAP recovery graphs here and we find GPA1, it's the purple line, and you can see that it really, really diffuses quickly within the plane of the membrane. So it's free. It seems to be relatively unconstrained. We did another protein, though. We tried one called AGP4. We tried a whole lot of different proteins, but we tried one called AGP4, and it turns out that AGP4, you bleach it, look what happens. 60 seconds later, the bleach spot hasn't changed at all, and in fact, it doesn't change. You can go back 30 minutes later, and you can still see the bleach spot. So if we look for AGP4 in this graph here, it's the black line right down here that just goes completely flat line like that. No recovery of fluorescence at all. So that tells us that fundamentally there's a difference in the way that proteins move within membranes. Some of them move very, very quickly, like GPA1 did, and some of them move very, very slowly, like AGP4 did. From there, as a scientist, you start to ask questions. Why is the one protein constrained? What holds it in place? Is it the cytoskeleton? Is it in a great big lipid raft domain? Does it interact with the cell wall if it's a plant cell protein? So these are the kind of experiments you can do with FRAP. I like this, and I spent a lot of time several years ago working with a new version of green fluorescent protein that's called photoactivatable GFP, PA, photoactivatable GFP. It does not fluoresce, in other words, it's black fluorescent protein, until it's first pulsed or activated with a short wavelength or UV laser. So we look down at a cell like this one here. We're looking at the inside of the cell. The GFP, the PA-GFP, is in the endoplasmic reticulum, but we can't see it at all until we fire the 405 laser. The 405 laser is this wavelength region here. You pulse the cell with this, and that activates the GFP so that it's excited normally with its blue laser, and it emits in its green wavelength range. So let's look at this movie here. This is 8 pulses separated by time. And look what happens. Little bits of GFP light up as they're pulsed with the 405 laser, and then they disperse very, very quickly. That was the scientific question that we had when we were doing this experiment. Do proteins that are within the endoplasmic reticulum disperse throughout the cell, or do they stay in place? Well, the answer is that they disperse, and very, very quickly. Look at that. After every pulse, the green dissipates through the cell. Pulse, dissipate, pulse, dissipate, and so on. We can do the same kind of experiment over here. This time we're pulsing continually within the yellow circled region here. The cell is this great big jigsaw puzzle piece shape that you see. We're pulsing continually within the cell, and over the course of about 10 minutes, the entire endoplasmic reticulum across that entire cell fills up with photoactivated green fluorescent protein. And what that proved to us was that protein that was here within the circle, when it was activated, could migrate throughout the cell, all the way up to up here. You'll see it turn green eventually. The green protein that's here had to have been down here when the laser pulse happened. So it's almost FRAP, fluorescence recovery after photobleaching, but in reverse, if you see what I mean, because rather than having a bright region that you bleach to black, you've got a black region that you activate to green, and then you watch where the green proteins go. Brilliant way to watch proteins moving from one organelle to another, for example. Okay, let's move on to the second experimental technique that I want to talk to you about today. This is one that has great utility in cell biology. It's used for determining whether two proteins are interacting with each other. And by interacting, I mean physically coming in contact with each other so that they can do their chemical reaction inside of the cell. In general, you might have something like an enzyme which binds another protein and causes a change in that protein. It might add a phosphorus to it or something like that, so that that protein is now activated and then does its job in the cell. So the big question in biology is, what are the roles of all of the proteins in the cell? And knowing what proteins interact with which other proteins is something that will really help us determine a protein's role. So the technique is called fluorescence resonance energy transfer. And I didn't put down the alternate name here. Some people say that since the technique was invented by Professor Forrester, that really it should be called Forrester resonance energy transfer. You don't need to really worry about that, though. If you just call it FRAP, the F can stand for Forrester or fluorescence. One fluorescent molecule can pass its emission energy to another as excitation energy. Stop reading right there. A fluorescent molecule, I explained this in the last lecture, has an excitation spectrum, a specific wavelength of light that excites it. So if you had two fluorochromes in a cell, and the emission, the light that comes from the first one, is exactly in the wavelength range that excites the second one, this one will emit light, which will excite the second one, and the second one will emit light. Do you get it? That's the transfer part of fluorescence resonance energy transfer. So here we have the first fluorochrome. It has excitation and emission, and we call that first fluorochrome the donor in a FRET experiment. We call the second fluorochrome, well, it would be the acceptor if the label here was in English, and the acceptor also has an excitation and an emission region. And the way this has been drawn, this one here is excited. That produces fluorescence emission. The fluorescence emission of the first one is right in the excitation range of the second one, and that causes the second one to fluoresce. And you can see that in this drawing down here. And the example we'll use pretty much throughout the lecture is of the FRET pairs CFP and YFP, cyan fluorescent protein, yellow fluorescent protein. Cyan is excited with a short wavelength laser, emits fluorescence emission, and if you were looking down at this cell, you would just see the cyan color because the laser that excites CFP is too short in its wavelength to excite YFP. So although both fluorescent proteins are in the cell, you don't see yellow fluorescence. But in this example over here, all we've done is we've put the YFP molecule rather slightly closer to the CFP. Look what happens. We turn on the laser, we excite CFP. It will emit some fluorescence, but some of its emission energy is transferred to YFP, and YFP emits. So in this case, where we say FRET has happened, turn on the short wavelength laser, which shouldn't excite the YFP, and the YFP glows. That's very, very curious. There's one absolutely important criterion for making FRET work in a cell, and it's the basis of the experimental technique. This can only happen if the two fluorescent tags are in very close proximity within the cell, i.e. within 5 to 8 nanometers of each other, and it indicates that the two proteins are physically interacting with each other. So here's my little scenario. All of the billions of proteins that are floating around inside of a cell are like this. They get close to each other, but not that close. They only get close enough for FRET to work if they physically interact. They occupy the same space. So that's two proteins interacting with each other. In the example that I've got in the picture down here, it's the fluorescent protein that frets with another fluorescent protein. So how do we get two fluorescent proteins so close together in the cell that they interact like this here? That's the basis of the experiment. In this real example of FRET experiment, the question is, do the proteins FRB and FKBP12 interact in a cell? Here's a little cartoon drawing of these two proteins. FRB is this little brown one here, and FKBP12 is this little brown one here. Now you notice something cute. FRB has CFP tagging it, and FKBP12 has YFP tagging it. In this situation within the cell where those two proteins don't interact with each other, we fire the 440 nanometer laser to activate CFP, and we see cyan fluorescence, but you'll notice there's no yellow fluorescence whatsoever. If we add rapamycin, which is an antibiotic, it binds to FRB, and it causes FKBP12 to associate with it in complex, just like that. So those two proteins now are said to be interacting with each other, and when they interact, they're close enough that their two tags also interact. So there is the CFP been brought right up against the YFP. Again, we fire the short wavelength laser to activate the CFP. We see a little bit of CFP fluorescence. You'll notice that the level has gone down, as indicated by the size of the arrows here. But what's cool is that we see yellow fluorescence, great big arrows there. So two distinctly different possibilities in this experimental setup. The question was, like it says in the top line there, are the two proteins interacting? You tag the two proteins with the FRET pair of fluorescent proteins, fire the short wavelength laser. If there's no yellow fluorescence, you say no. The conclusion is those two proteins don't interact with each other. But if you fire the short wavelength laser and you see that the two proteins are both fluorescing, but in particular you see the YFP fluorescence, you say that means that the proteins are definitely interacting with each other. And here's just a picture to illustrate that. GFP and RFP used in this case is the FRET pair. The only requirement for a FRET pair, I'm going to go back a little bit, the only requirement for the FRET pair is that the emission of the first one overlaps with the excitation of the second one. So the famous FRET pairs, the first one was CFP and YFP, like illustrated in that picture, but it turns out that GFP and RFP also fret with each other quite well. And people are developing new pairs of fretting fluorescent proteins all the time. So GFP and RFP are used. GFP is attached to one protein and RFP is attached to another protein. The question is, do those two proteins interact within the cell? If the two proteins don't interact, you fire the shorter wavelength laser to excite GFP, you see GFP fluorescence, but very little or no red fluorescence. And you say your conclusion is, okay, those two proteins don't interact with each other. But if you fire the short wavelength laser and you see red fluorescence being emitted, you know that the two tagged proteins are interacting with each other. So we conclude proteins A and B, which were fused with green and red fluorescent protein respectively, do interact with each other in the cell. Here's a nice technique which sort of follows on directly from FRAP. This is called FLIM, Fluorescence Lifetime Imaging. So do you remember when I talked about fluorescence, I talked about how a molecule is down here at its ground state. It's excited by the input of energy, which is the lasers in confocal microscopy. That causes its electrons to jump up to a higher energy shell, which is up here. And those electrons fall back to ground state with emission of heat, but also with emission of fluorescence. So in this example here, we've got a fluorochrome that is being excited with blue light, and it's emitting some green light. Well, you can measure the brightness by looking at how much emission there is, but there's something else that you can measure, and that is how long the fluorescence lasts for. In other words, instead of having a continual input of energy that causes continuous fluorescence emission, what if you just pulsed one very quick, one short pulse of energy to stimulate this excitation, and then you watched how long the molecules stayed fluorescent as they drop back to ground state. Remember, when you put a pulse of energy in, you're exciting a whole pool of fluorescent molecules, millions of them probably. They jump up, and then the fluorescence decays away. Well, you can pulse, and you can measure how long it takes the fluorescence to decay away. And this is in the nanosecond domain, by the way. Maybe 2 to 3 nanoseconds, something like that, is how long the fluorescence from a single fast pulse of a laser acts. Well, it turns out, and this was an amazing discovery, when two proteins are interacting with each other, like these two did back here, FRB and FKBP12, when two proteins interact with each other so that one is passing its resonance energy to the other, instead of all the light going as fluorescence, some of it goes to the acceptor molecule, which then becomes fluorescent, and that fluorescence decays away, and you can measure the rate of decay there of this original fluorescence on this side. Well, what happens if there is FRAP, if the two proteins are interacting with each other, the decay is much quicker. So we can look at the amount of time fluorescence lasts here, the lifetime of the fluorochromes, and we can look at it here and we can say, wow, when we've got those two proteins labeled, the fluorescence lifetime is much, much shorter. So it's another way that we can determine whether proteins are interacting in the cell. Here's a real experiment we did one time. We did an experiment where the fluorescent protein wasn't interacting with anything. The lifetime of the GFP was 2.4 nanoseconds. And when we had the GFP interacting with another, when we had the protein that the GFP was tagged to interacting with another protein, the fluorescence lifetime dropped to 1.9 nanoseconds. I remember being there on the day doing the experiment. This was down at the Central Laser Facility down near Didcot in Harwell in Oxfordshire. It's this amazing building that has all of this high energy physics. They've got big giant lasers, giant microscopes everywhere, and these guys were doing this FLIM experiment with us. And I saw this result, the one that's indicated down here, and I was underwhelmed, to say the least. I looked at it and I thought, okay. Man, the people that were running the facility, as soon as they saw the result, they went, whoa, and they started high-fiving each other. And I thought, do you mean that's something, that drop from 2.4 nanoseconds to 1.9 nanoseconds? They said, you don't even understand. That's absolutely amazing. That's a super strong protein interaction. And the lesson I got from that was just how small the things are that amuse physicists. But it amused me greatly too a year later when we got the paper published. So there you go. That's FLIM. Bimolecular fluorescence complementation, and I've said also known as the split fluorescence protein technique, or the "this is beautiful, why didn't I think of it" technique. When I first heard of this one, I thought, that is so cool. That's amazing that that works. So you remember when I told you last week about how we get fluorescent proteins into cells to tag organelles. We fuse them. We make what's called a fusion protein. You can see here that protein X, for example, may be a bit of a linker, just a few amino acids in between it and the GFP. And when I explained this last time, I said that this protein here will go in the cell where it's supposed to go based on targeting information that its sequence contains, and it will take the GFP along as a tag. And I showed you examples, the Golgi apparatus lit up with green fluorescent protein. Well, in this bimolecular fluorescence complementation technique, they go one further and they do something that is amazing. They take the GFP gene and they split it in two. They label, or they designate, the one part of the construct as N-GFP. In other words, the N-terminal half of the green fluorescent protein sequence goes in that construct there, fused to protein X. And the C-terminal part of the GFP molecule goes in this construct here, fused to protein Y. Then your question is, just like in a FRAP experiment, are proteins X and Y interacting with each other in the cell? Look what happens. If X and Y interact, they come right together. Remember this thing here. That brings the N and the C terminals of GFP together, and they fluoresce. Now that's cool. That's an absolutely brilliant way to conclusively demonstrate that X and Y are interacting with each other. What amazes me even more so is that if you've got half a protein here, the N-terminal region, and the other half of the protein here, the C-terminal region, you bring those two together, they actually knit together and they make a fully functional protein that's capable of emitting fluorescence. But it really, really works. I've seen it in action. It's so amazing. So the gene coding the fluorescent protein is split into two parts, fused to two different proteins, X and Y. If there's no fluorescence, that could mean that X and Y don't interact with each other, but very definitely if there is fluorescence, that means that X and Y interact with each other, and you've just proven part of a biological mechanism. Here's a real-life example of a very, very cool BiFC experiment that goes a little bit beyond what I've explained to you. In this case here, they're expressing three things inside of the cell. They're expressing the N-terminal domain of YFP, the front half of the protein, the C-terminal domain of CFP, and the N-terminal domain of CFP. So, if proteins B and Z interact with each other, like has been done here, the N-terminal of CFP will fuse with the C-terminal of CFP, and you'll get fluorescence inside of the cell, as indicated in this picture down here. So that works. That's really good. If the N-terminal of YFP fused with the C-terminal of YFP, you would get YFP fluorescence, but that's not what's going on here. Look at this. You don't need to put the C-terminal of YFP in, because if the N-terminal of the YFP protein fuses with the C-terminal of the CFP protein, that gives yellow fluorescence. So if A and Z interact, like they're doing here, you get yellow fluorescence, even though half of this molecule is the C-terminal domain of the cyan fluorescent protein, you get the yellow. That picture there is the merge of the two things. So when B and Z interact, we're localized to these little dots within the nucleus. Those might be the nucleoli. And when A and Z interact, we're localized within the nucleoplasm outside of the nucleoli. So that's really, really interesting. Just a little comment on the fact that you get yellow fluorescence even though you've got the C-terminal of the CFP protein. If you remember, Roger Chen's group made YFP and CFP by making small mutations in the wild-type GFP sequence. Well, wild-type, or eGFP, is very, very similar to YFP and to CFP. The mutations that cause the color shift are very definitely in the N-terminal domains. So the C-terminal domain is unchanged. This could just as easily say CYFP here. So that's a really, really cool experiment. They showed a couple of different pairs of protein interactions, and they've shown that when the different pairs, when the protein Z interacts with A, localization is within the nucleoplasm. When the protein Z interacts with protein B, localization is within the nucleolus. Total internal reflection fluorescence. This is a way to monitor things moving around within cells. The other techniques that I've told you about so far, FRAP, photoactivation, FRET, FLIM, and BiFC, all done with a confocal microscope. TIRF is actually not done with a confocal microscope. It's done with the very same sort of research microscope, but it doesn't require the confocal pinhole, and it's because it operates with a different sort of light. Here is the light coming up through a slide and illuminating a specimen, which is a cell in this case, that has green fluorescent particles in it. It says that this is epifluorescence. If it was confocal microscopy, of course, the pinhole would give a thinner band of fluorescence somewhere within the cell, wherever you were focused, but you would get a lot of green fluorescence coming from the cell. People were so frustrated because they couldn't get a thin enough view, just what goes on at the plasma membrane where the slide contacts the cover glass. To do TIRF, total internal reflection fluorescence, what you do, instead of firing the light straight up to illuminate the cell from underneath, you fire the light at the slide at a very steep angle. Well, what happens, because of the glass interface that's right here in the cover slip, the light reflects away. So this is the incident light coming in, the little blue line here, and reflecting. That's the total internal reflection. That reflection, though, creates what's called an evanescent field. And the evanescent field is exciting light, which excites fluorescence in the fluorochrome that's right there, only within a very, very thin region. In other words, only within the thickness of, let's say, the plasma membrane and maybe the very, very underlying region of cytoplasm there. So you don't have a whole lot of fluorescence from the whole rest of the cell up here at all to contend with. So using TIRF, fire the light at a steep angle, see fluorescence in a very, very thin layer, and it lets you see very, very small things indeed, right down to single fluorescent particles, single fluorescent proteins. So that's why we use TIRF. Let me show you a real-world example of this. Two different proteins are fused with GFP and imaged using TIRF. The GFP fluorescence appears as white in these movies. You barely see it. What we've done is we've turned on tracking software so that during the time series of the movie, a protein that's fluorescing in the plasma membrane will appear and it will move. And we track its movement and we draw little pink tracks to show where the protein is. So each circle that appears and dances around a little bit is an individual fluorescent protein that's tagged to a plasma membrane protein. It dances around within the membrane for a period of time and then it disappears. These movies were the most exciting bit of research that I've ever done in my lab. This was just one of the coolest things, and they are, truthfully, some of the most boring movies you could watch. You should see how boring they look if you don't turn on the circles and the little pink tracks. In any case, let me just stop that so you don't get hypnotized. That protein that we were looking at is called LTI6B fused to GFP. LTI6B is unconstrained. It moves around very, very quickly in the plasma membrane. And we know that already from doing FRAP experiments where we bleach the membrane, and over the course of a minute the bleached area becomes bright green as the proteins move back into it. But when we're doing a FRAP experiment, we're looking at millions of molecules of the fluorescent protein. In this case here, using TIRF, we can look at the individual molecules and we can see how they move. We did the exact same experiment on a different protein. This time we fused one that's called FLS2 with GFP, and watch the difference when you see the movie. These two movies are recorded at the exact same frame rate. So look at all the activity in the protein at the top. In fact, let me go back to the start of the movie. Look how fast those appear and how fast they move around and how big the tracks are. You can see the tracks here, for example, of one of those proteins are quite long. Meanwhile, while all that's going on in the top video, look what's going on in the bottom video. FLS2 is clearly really locked in place. It appears in the plasma membrane, but it sits still. And our question was, what is it interacting with? Why is it constrained in its motion? The bigger biological question was, FLS2 is a defense protein. It defends the cell from pathogen attack. Does a cell just willy-nilly put proteins out into its membrane and let them diffuse around freely, in other words, at random, or does it place them very specifically, like sentries at guard posts? And it seems like the answer is that the protein is placed very specifically in the plasma membrane, and once it's there, it doesn't move. We did the same kind of experiment at that same time, studying the protein FLS2, again, the one that I showed you in the bottom video on the last slide. We wanted to see if the lateral mobility of the protein changed if we added its ligand. So it is a receptor. I told you that it protects cells from pathogen attack. It's a receptor, like a satellite dish, on the that has its receptor on the outside of the plasma membrane. What it does is it senses proteins, little chemical signatures that come from the surface of bacteria, in this case from the flagellum of bacteria. So what we did was we added a little bit of that flagellin protein to see whether that changed the motion of the FLS2 protein in the membrane. So here we go with no flagellin 22 added. Watch the circles, see the pink spots, and you see the protein in the membrane. Okay. Then we added flagellin 22, and we added it at a couple of different levels. This simulates pathogen attack. And you see circles appearing. And you see little tracks for the molecules as they move around in the membrane. Hello, there it is. The question is, are the tracks here different? Is the protein faster moving than here when it's actually sensing bacterial attack? The only way to get at that, by looking at the two pictures, I mean, if you play the two movies at the same time, it does look like things maybe move around a little faster here, but we don't know. Very often when you do experiments, any of the kind of experimental techniques that I'm talking about, it's a little bit subtle. Is there a difference or not in what's going on? And that's where we turn to statistics. Let me just stop these. We turn to stats and we use all sorts of image analysis to determine what's going on. And in fact, what we found was that the control moves at this rate here. I'm not going to explain this MSD calculation. And when we add the flagellin 22, this was 10 micromolar in the movie, I'm showing you the movement of the protein very significantly slowed down. So as soon as the receptor binds the flagellin, something happens to it in the plasma membrane. And we're continuing with that research now. Okay, that's the end of the first half of today's lecture. If you go back to Moodle, you'll find the second part of the lecture.